So good afternoon everyone, and welcome to the Ensembl 83 release webinar. My name is Ben Moore and I am an outreach officer for Ensembl. So I'm part of a small team that's responsible for delivering workshops and webinars like this one. We also create and maintain all of the online help and documentation that you might have used before. And we also answer your questions via the help desk, and we're in charge of all of the social media. So Facebook and Twitter. If you follow us online, then you might have seen us there before. My colleagues, Emily and Helen, are both here today, and they're going to try and answer any questions that you might have about the webinar in the comments box that you have available in the software. So I want to start by giving you a brief background to genome browsers, and in particular Ensembl. So I'm sure many of you are aware that Ensembl is a genome browser, and it was first conceived of in 1999, I think, when the first human genome was about to be published. But the first genome actually to be sequenced was a bacteriophage in 1977 by Fred Sanger and his team, and it was a relatively small genome. It was only 5 kilobases in size, but it was a massive breakthrough at the time, and it really paved the way for more and more complex organisms to have their genome sequenced. That eventually culminated in 2004 in the publication of the first human genome sequence, and this was 3 gigabases in size, so this was much bigger. And it was, again, it was a massive achievement at the time. But since then, there have been even more technological increases that have allowed more and more sequencing data to be produced. So more organisms are having their genome sequenced, and I'm sure this is going to continue in the future. But also there are large projects such as the 1,000 Genomes Project and the 100,000 Genomes Project, which are aimed at sequencing the genomes of a large number of individuals within subpopulations and within specific species. So all of these sequencing efforts are resulting in the accumulation of a large amount of raw sequencing data, which without any context or annotation seems to be quite meaningless. So this is where genome browsers, and in particular Ensembl, they aim to process all of this data. They try to make it accessible and meaningful. And Ensembl does this in a variety of ways. So for all of the species that we have in Ensembl, we have genomic assemblies, which have been automatically annotated with genes from our automated gene annotation pipeline. We also have a large amount of variation data. So we have small and large scale sequence variants with phenotype associations where that data is available. We also have comparative genomic analyses. So we have whole genome alignments and gene trees, for example. And finally, we have regulation data. So we draw in data from a large variety of sources to predict potential regulatory features such as promoters and enhancers. So Ensembl tries to bring these different arms of genomic annotation together with a number of features. So firstly, we have what we call gene builds using this automated annotation pipeline for the species that we have hosted on Ensembl. And these produce gene sets that we compare pairwise with from species to build gene trees. We also have our regulatory build, which draws in data from sources such as GENCODE. We have, as I said, a large amount of variation data displayed in Ensembl, but you can also analyze your own variation data using the variant effect predictor, or the VEP, which we call for short. You can upload your own data to Ensembl. You upload your own data files and view them in the web interface. And you can also export Ensembl data using Biomart. You can export large amounts of data in that way. And you can also access Ensembl programmatically through the REST and the Perl APIs. And the great thing about all of this is that it's completely open source. So all of the data, all of the code and the software that we have in Ensembl is completely open source for you to use in downstream applications that are suitable for your research. So we have a large number of species in Ensembl, which is primarily dedicated to the vertebrates. And we try to cover a broad taxonomic space. So but we also have agriculturally, medically, environmentally important organisms. So you can see we have the great apes, including humans. We also have a number of primates and rodents. We also have other placental mammals. We also have marsupials and monotremes, you can see there in the top right-hand corner of our radius. And then we also have some birds, reptiles, fish, and finally some non-vertebrate chordates such as the lamprey and the sea squirt. Finally, we also have 3 non-vertebrate species hosted in Ensembl. So we have C. elegans, the fruit fly, and Saccharomyces cerevisiae, which are all hosted because of their importance as model organisms in biological research. So Ensembl works in a release cycle mechanism. So our last release was Ensembl 82 in September of 2015 this year. And since then, we've included a large amount of new data, new software updates, and we've updated interfaces and usability. And we package this all together and release it in a new release that we mark that we've marked as Ensembl 83. And this was released at the beginning of last week, and is the reason for this webinar. The reason why we do this is to make everything very traceable, so you can access all of the old data and software that we used in the old Ensembl releases via our archive sites. And this means that if you've read papers that were published a little while ago, or you've made notes or have data in your lab book from months or years ago, then you can trace back to see where this may have come from in the Ensembl archive sites. So what's new in Ensembl 83? We have updated gene sets and annotations for human, mouse, and rat. We also have variation and phenotype data updated for several species, again including human, mouse, and rat, but also including many other species. We have a new exact variant set and track that I'm going to show you how to use later. And we also have variant table filters that have been updated to allow you to narrow down the variants that you're looking at in the variant table more accurately. We also have the Manhattan plot for linkage disequilibrium that you can look at. And we also have an improved image export function that I'll finish with the webinar by showing you that. This list is by no means exhaustive. There's many more updates. This is just the headlines of our updates. If you want to see all of the updates, then you can use the link at the bottom of this page, or you can click through to all of the updates via the Ensembl homepage. So as I alluded to earlier, all of the species in Ensembl have their genomes annotated automatically using our automated pipeline. But we also have manual annotation for a select number of species. So we have manual annotation for human, for mouse, rat, pig, and zebrafish. And this manual annotation is performed by the Havana group. So the automatic gene annotation is a genome-wide determination of gene models using the automated pipeline, and it's based on experimental data. So the experimental data that we use is sequencing data that's being deposited in the International Nucleotide Sequence Databases, ENA, GenBank, and DDBJ. Also, protein sequence databases are used in the automated pipeline. So we have Swiss, Prot, and Tremble, for example, that we use data from to annotate our sequences. And finally, we have manually annotated proteins and mRNAs from NCBI-RefSeq. So the GENCODE gene set is the name of the gene set for either human or mouse. that is made up of the merged set of both Ensembl automatically annotated genes and Havana manually annotated genes. And the GENCODE gene set is the default set that's used by ENCODE and other major projects such as the 1000 Genomes project. So in Ensembl 83, we have updated gene sets for human, mouse, and rat. For human, we've updated to GENCODE 24, and we have new assembly patches as well. For mouse, we've updated to GENCODE M8 annotation, and for the rat, we have an updated gene set, including manual annotation from Havana. As I mentioned earlier, we have a large amount of variation data that's displayed in Ensembl, and this broadly comes in two flavors. So firstly, we have small scale variation data, and this is variants that affect one or a very small number of nucleotides. And these generally include single nucleotide polymorphisms, for example, or small insertions and deletions. Secondly, we also have large-scale variation, for example, copy number variations, or large deletions, duplications, or insertions or translocations. And this variation data is displayed in Ensembl through a variety of sources. I'm just going to draw your attention to a few of them. So firstly, we have dbSNP. We also have OMIM, which is online Mendelian inheritance in man, and DGVA. But obviously, there's a large number of variation sources that we use, and you can find all of the variation sources using the link at the bottom of the page. As I alluded to earlier, where available, all of the variation data that we have is associated with phenotypes for the different organisms that they represent. So in Ensembl 83, we have updated the human phenotype data from the NHGRI EBI GWAS catalogue, also from OMIM and ClinVar, UniProt, OrphanNet, the Cancer Gene Census, and GOA, which stands for Gene Ontology Annotation. We also have GOA data updated for cow, dog, sheep, zebrafish, chicken, macaque, and turkey. We also have OMIA, the data, which stands for Online Mendelian Inheritance in Animals, for cow, dog, horse, and sheep. We've also updated phenotype data for rat from the rat's genome database, and also for mouse from IMPC. And finally, we have animal QTL phenotype data for cow, horse, chicken, pig, and sheep. As I said earlier, we have brought in variation data from the DGVA, which is a repository that provides archiving, accessioning, and distribution of publicly available genomic structural variants for all species. And we've updated, we have new and updated studies from DGVA in Ensembl 83 for human, mouse, cow, horse, dog, and pig. We also have the latest sequence variants from dbSNP 145 for chicken and pig. And we've also imported the variant IDs from dbSNP 145 for gibbon. Finally, for variation data, we also have the new EXAC variation data available. So EXAC is a coalition of investigators, and they aim to aggregate exome sequencing data from a wide variety of large-scale sequencing projects, and to make summary data available for the wider scientific community. And we have the new EXAC track available on the website in the web interface that you can add to your views. So now I've told you about all of the new variation data that we have available. I'm going to give you a quick demonstration of how you can access all of this data. From the Region in Detail page, you can access this variation data by clicking on the Configure This Page button to the left-hand side of the screen. And this will bring up a pop-up window where you can choose to see all of the different variation data available by selecting the drop-down, the expandable menu to the left of the pop-up window. As you can see here, I've selected the exact data, but also the 1,000 genomes data and all phenotype-associated data. And you can obviously choose which data that you'd like to see by clicking on the empty boxes. Once we accept the data that we wanted to see, it's displayed against the genome assembly. So here you can see the new exact variant track, and you can view these different variants by clicking on, zooming in perhaps, and clicking onto the individual variants and getting more information about them that way. So as I mentioned earlier, we have new variant table filters, and we basically just have a wider range of filtering options in the variant table. And these filters include consequence type, which we had before, but also minor allele frequency, SIFT and polyphen scores, and clinical significance, among others. And it just allows a more flexible way to narrow down the variants from the long list of variants in the table that you might have. So I'm going to use BRCA2 as an example here. So from the Gene tab of BRCA2, you can click on the variant table option in the left-hand side menu, and this brings up the variant table as it has done before in previous releases on Ensembl. If you scroll down, you can see the long list of variants in the variant table, but you can also see the filter options at the top of the page here. So you can choose to filter the variants by global minor allele frequency, and you can choose the frequency that you like by using the drag function in the box here. You can also filter by SIFT scores, polyphen scores, and sequence consequences that we've had before. You can turn on or off the different sequence consequences by using the green buttons. And there are other filter options, so for example, clinical significance or location you can use. So as you can see here, I've filtered the variants by polyphen score. And so I've selected to only see variants that have a polyphen score between 0.9 and 1. And I can use multiple variant filters all at once. So if I also wanted to see only variants that had also a SIFT score, then I could choose two different variant filters at once. And again, you can turn these filters off by clicking the green cross next to the relevant boxes. We also have the Manhattan plot track available, and this is a new linkage disequilibrium track that displays population-specific LD values for linked variants surrounding a focus variant. So linkage disequilibrium is calculated in two ways. So we have the R squared and the D prime values, and they're calculated for each variant in relation to the focus variant, which in this case is RS1333049. And the R squared or D prime values are calculated for each variant in comparison to the focus variant and plotted along the track between 0 and 1. So you can find the Manhattan plot track by accessing the variant information pages. So we had our focus variant that I mentioned before, RS1333049, and you can find the Manhattan plot track by clicking on the linkage disequilibrium link to the side of in the menu to the side of the screen, or through the thumbnail in the main page. This will bring you to another page where you can choose the Manhattan plot track that you want to view for specific subpopulations. So I'm going to show you the Manhattan plot for the African, Caribbean, in Barbados population from the 1,000 Genomes project, and you can access this by clicking on the link just here. And then the Manhattan plot track will be displayed against the genomic assembly, and you can see this is the example that I took from the previous slide, and it's displayed against the variants, the corresponding variants above. So finally, I just want to finish up by showing you our new improved image export function. So we have both a presentation and a journal or poster image export function. So the presentation function alters the exported image to be clearly visible on projectors if you're perhaps giving a presentation. And our journal or poster print options produce enlargement, so a ×2 for journal or a ×5 enlargement for print purposes. And basically, the improved image has adjusted colors to improve the contrast and increase line breadth for viewing images from a distance. So you can export images in Ensembl 83 just in the same way as before by clicking on the Export This Image icon that's to the top left-hand corner of any image in Ensembl. And that brings you with a pop-up menu here that you can see. And you can choose the different formats for the exported image that you want. So you have the journal/report, a poster or presentation options, or also web and PDF files that you can export. And here I just have a side-by-side comparison of the presentation image exported. So on the left is just the normal web export that we had before, but on the right is the presentation exported image, where the colors are slightly darker to improve the contrast, and it just makes it a little bit clearer in a well-lit room. So just a few things before I finish up. We'd like to tell you about our Ensembl workshops. So we can teach an Ensembl course at your institution for free, except the train's expenses. If you'd like to contact us via the Ensembl help desk, or you can email me directly, and we can come and give either an introductory browser course, a half to 2-day course on the Ensembl browser aimed at wet lab scientists, or we can give a more in-depth API course, which is between a 1 to 4-day course on the Ensembl Perl API, and this is more aimed at bioinformaticians. You can learn more about Ensembl by using our online tutorials via the web link that we have there, and all of the webinars, and we also have a number of recorded demonstrations on YouTube and Youku. And finally, as I've mentioned before, there is the Ensembl help desk. So if there's anything that is not clear about the webinar perhaps today, or if you have problems or queries when you're using Ensembl, then you can email helpdesk@ensembl.org, and we'll try and help you there. So this is the Ensembl team, plus or minus a few people, as this is a few months old now, but you can also keep up to date with us by following us on Facebook at facebook.com/ensembl.org. You can also follow us at Ensembl on Twitter, and we also have a blog, www.ensembl.info. So I'd just like to acknowledge all of the members of the Ensembl team who have put a lot of work into the latest Ensembl release, and all of our funding bodies who have made all of the updates and work possible.